Now that Bay's here, we can get started. Good timing, Bay. All right, well, welcome. Good afternoon and welcome. Looks like these clocks have not caught up with our daylight saving time yet, but it actually is a little after noon. And we're very happy to have Amina Qutub, maybe close, here from Rice University. She's a professor in the Department of Bioengineering there. She received her PhD from the University of California at Berkeley in bioengineering, and then did a postdoc at Johns Hopkins University, and knows a lot of the people that we work with and know at Johns Hopkins, and including Ray Winslow, who's a member of our external advisory board for our NIH Center. So we're very happy to have her. She really touches on a number of different areas that we also work in. So it'll be very interesting to learn about her work. She's going to talk about visualizing human cell communications. So please join me in welcoming Amina to Utah.
Thank you, Chris. Thanks, Chris. It's been a joy already this morning to be at the Ski Institute and see the interdisciplinary work that's been going on so far and where you're going with some of the tools. My lab is interdisciplinary, as you can see from the website, and what we're interested in most is understanding how cells communicate during growth. But I want to take you back a little bit about showing where our inspiration is coming from. And it actually, for me, comes from the humble P. And this P actually was a model system for one of the first people I think of in data science applied to biomedicine, and that is Gregor Mendel. And in fact, over the course of 7 years, he laboriously took the time to characterize the shape and structure of 28,000 pea plants, characterizing from the different traits from a perspective of trying to come up with a rule that could potentially explain how these traits were showing up in different generations after generations of the peas. When you think about that, that's one of the first applications of taking a data-driven approach to come up with a long-standing theory. And it wasn't really appreciated until decades after his work, and actually many people here have built off of it in beautiful ways. And it's interesting to see applications of this work today and in the future. My work takes an approach where we almost take the peas out of the pod. In a sense, we're taking the human cells as our model organism of what the human is doing. And similarly to Gregor Mendel, we're characterizing them. And we're characterizing them from many of the omics perspectives that are used commonly in bioengineering. But also we're interested in their behaviors and what they do during processes of growth. And when I mean process of growth, we're interested in two different sides of that coin. And you can think of growth, malignant growth during cancer. There's too much growth. You want to understand how this process happens to the cells. And on the other hand, in some cases, you want to regenerate tissue after a stroke or after an injury. So you want to understand how cells are not being able to regenerate. And the molecular mechanisms, the common molecular mechanisms are also the starting point for where I started this work. And that both growth processes involved a process called hypoxic response, or how cells respond to local limited oxygen levels. I want to first start a little bit with where we're going in these two domains of my work, and then dive into an overview of the projects. And feel free to interrupt me at any time during this talk. On the cancer applications, we're interested in following up on what Weinberg and Hennehan had done for many decades, and they had published seminal papers about these hallmarks of cancer. And they had identified these hallmarks through a meta-analysis of the literature and their own experimental work. What they hadn't done is followed up on it in a quantitative way. And I was interested in understanding, we've been interested in understanding, is are there quantitative rules or hallmarks behind a way a cancer cell can become cancerous? And for this, we're specifically focusing on cancers of the blood and bone marrow, or leukemia applications. On the other hand, on the regenerative medicine applications, I've interested in applications towards the brain and the neurovasculature. So there, we're trying to understand what are mechanisms of healthy tissue regrowth. So briefly on the cancer side, there's a lot of publications that have been out recently on the deep methodology and the mathematical background behind what I'm presenting today. I'm not going to go into those details at this talk, but welcome any questions on any of things that we've published or any on the methods or tools that are out there. What I am going to go through on this talk is an overview of the approach we're taking and where is it getting us. So acute myeloid leukemia, the reason in part why I was focusing on this work is because when I first started at Rice, I had met our now long-term collaborator, Stephen Kornblau. He's a clinician at MD Anderson Cancer Center working on acute myeloid leukemia. And we met at a conference in the mountains in Colorado on hypoxic response, or how cells respond to low oxygen. But he was posed with this problem of how to handle all the data he was working with. And in particular, the data for leukemia is extremely heterogeneous. It's a disease of the blood cells or the myeloid stem cells in the blood that instead of becoming normal red blood cells or white blood cells, they stop at a earlier stage in differentiation and accumulate quickly. And at that point, they start to accumulate in the bone marrow, and they take on characteristics of the cancer. It's a disease of more prevalent in the elderly, so people over 65 and who are male are more likely to have acute myeloid leukemia. But what's really interesting about this from a data science and computational perspective is that it's one of the most heterogeneous diseases out there. In fact, a lot of people describe it as not one disease, but as a collective of many diseases. And the approach so far to catalog or to understand this complexity has been to look at the cytogenetics. So if you ask a doctor now, when a leukemia patient comes into their office, how would they classify them? What's the most robust classifiers for their disease? What they'll point to is cytogenetic categories. So are these patients under what's called a favorable cytogenetic category, intermediate or unfavorable? And those are groups by chromosome abnormalities or differences at the chromosome level. But what it's not getting at is greater complexity downstream, epigenetic effects, and even further downstream, the proteins that are actually responding to the local microenvironment, along with changing the cell expressions patterns and how they're behaving. So what we were interested in doing is, can we take what we know about the proteome and ask two questions? Is it true that we can think of two hypotheses underlying the proteome for acute myeloid leukemia? The first one being that it can be characterized by a succinct set of protein expression patterns or rules that govern every cell of patients. And the second is, can we use these rules or this signature and look at it from a global perspective or a roadmap of how you might develop cancer, leukemia? So the data that I was getting and our collaborator had been collecting over the course of several decades is painful data to get. It's a bone marrow biopsy. So you aspirate bone marrow from the back of a patient, and you collect that, and you do an omics analysis on it. And one of the methods we do is reverse phase protein array analysis. It's a dual antibody-based method, and the outcome of it is we get expression levels for 232 proteins, and including some of the phosphoproteins and their main protein. And also we get 80 clinical covariates. And those things are things like age, or whether somebody had a prior malignancy, or whether they had an infection. This is the data set, and this is the data set that we got when we first looked at it. And to me, I don't know if you see old TV screens anymore that used to have a lot of noise in it. To me, that's what it initially looked like. And this is looking at the data where you have all the proteins on the x-axis, and then the patients are on the y-axis. And it's just a hierarchical clustering of the expression levels from high to low, from blue to red for the highest levels. So every patient is one of these columns, thin columns, and then you see the expression for every protein for the patient. But this was looking at the data in a way where all the data was considered flat. There was no hierarchy to the structure of the data. And we know in biology there's hierarchy, that one protein can be a transcription factor and another protein can be important for its phosphostate. And so what we wanted to do is identify two things. There were two computational challenges among many others that we saw we could tackle. First was, how do you know the optimal number of patient groups? It seems like a straightforward question, but it's really a tricky one. How do you know how to group patients in this high-dimensional data set? You do have a limited number of patient samples, and you have high-dimensional data. So clustering itself is an art. So we developed a number of methods to do this optimization, and a couple of them have been recently published, shrinkage clustering and progeny clustering. I'm not going to talk about that work here. What I am going to talk about is what we're doing on the pattern discovery approach. What you also want to know is how do you discover signatures from this data, which is so complex and high dimensional, and how do you integrate known biological knowledge into those patterns? So to do that, we first stepped back and said, well, it's okay to look at these proteins as individuals, but as a collection, they're more important. It's almost like looking at all the cities in the US as individuals without looking at the map of how they're located next to each other. Is there something more interesting about how these proteins are interacting with their neighbors that gives us something better information about how to classify patients for therapy? And we did this by first starting with doing a functional annotation. So example, you can think of every one of these as proteins, and on their own, they might be informative if they're high and low, but together they might give you a rule, or a word, like nice day, that would give you something that tells you something about their function. So that's what we started with, and we used culling from literature to know how to define these functional groups, and then also the expert's opinion, the doctor's opinion on how to define these 31 functional groups. From that, then the data showed us the functional patterns of the expression of proteins involved in those groups. And from these functional groups, you can get an example of, this is another heat map. Our clinical collaborators love heat maps, so this is one I'm showing you in this form. Again, every patient is one of these thin lines, if you walk down the x-axis, and each of these are proteins, on the y-axis, and each of these are proteins. And this is the SMAD protein family, and you see some phosphoproteins, and the color corresponds to very high expression being red, and the dark blue being low expression. And you can see 5 patient groups or functional groups that were identified through this method. What we then do is you look at each one of these as a network. So these groups are telling you about a pattern in the structure of the data in a collective. It's not just one protein's high or one protein's low, but the functional grouping of them is important. The edges or between every 2 proteins is defined by Glasser regression in combination, graphical lasso, in combination with a meta-analysis or a computational analysis of culling from what is available from known protein-protein interaction databases. From these, you can then correlate it to outcomes. So this is a survival curve at the very beginning. All of the leukemia patients are alive, and every tick mark is when somebody is leaving the study, and when you go down the y-axis, you can see somebody is dying off in the survival curve. Time is in weeks on the x-axis. So this allows us to look at which networks or pattern of networks is responsible for the worst survival and the best survival, and in turn identify potential new targets. So for us, targets would be things like SMAD1, which is a protein we know is involved in TGF-beta signaling, and it leads to greater growth of cells and limited apoptosis. In turn, there's another way that same signaling pathway can be interfered with through SMAD2 phosphostate and its changes, which lead through another different type of aberration in TGF-beta signaling, which in turn leads to proliferation of cells. So this is one of the 31 pathways that I'm showing you. You can do this also for the pathway of interest that I first started with looking at, and that is how cells respond to low oxygen or hypoxia. And here, again, it's similar. These are 2 curves which look at remission, so the amount of time a patient is in remission and hasn't relapsed in leukemia. And what you're seeing again is the patterns of the functional groups, and we can pick out which are the patterns that correspond to the best prognoses and which are the patterns that correspond to the worst prognoses. And now we're going back in the lab and targeting those proteins through a synthetic biology approach where we can manipulate not just their on-off states, but their relative states to each other in cell lines of these patients, correlated to these patients' data. So from what I've shown you so far, you get functional groups and the patterns of expressions in those functional groups. But we wanted to take it a step further because that doesn't allow us just alone to compare across the board how the cancer is behaving across many functional groups. So we combine the functional groups into what we call a constellation. And a constellation is represented through this binarized heat map or cluster graph, which I'm showing you. So what we did is every square, we've asked for an individual patient, and again, a patient is one thin line as you look down this chart, like here. And each one of these corresponds to whether a patient falls into that category of the network or not. So it's 0 or 1, 1 if they do, 0 if they don't. So for hypoxia, there were 5 signatures, and each one of these signatures is a network. So for one patient, they would fall into only one of those 5 signatures. And in turn, for the SMAD group, they'd fall into one of those 5 or more signatures. And this gives us then a lookup table for how do patients fall or classify into these protein signature groups. It also gives us which are the protein groups that are functionally connected during the processes of leukemia or stages of leukemia. So for us, SMAD5 is, in this cohort of patients, is connected to the STAT pathway group 4 and T-cell group 4, as an example. And then in turn, we ask the question of, are there signatures that, these signatures, do they correlate to outcomes? And they do. You can see it here. We've divided our outcomes, just like you divide cytogenetics into favorable, unfavorable, and intermediate. We divided the protein groups, which are targetable, into different categories. And what we're showing here is that the protein groups alone add more information than the current state of the art, which are cytogenetics and white blood cell count and age. So they can give you information independent of those current prognoses. Beyond that, we wanted to step back. We wanted to really get at hallmarks of cancer. So how do you get at that through this type of computational analysis? One of the ways to do it is to look at each signature and ask what's happening at every level of these proteins. So we built up a decision tree. So at the very top of the tree, there's the most information. At the very bottom of the tree, there's the least information. So the best classifier is at our top. So this means if we know a patient's in PI3 kinase group 1, we know that's very informative for whether they're going to have a good outcome or not. So we walk down the tree to find each patient group for outcome. So for the beginning, the worst prognosis group, the group that I highlighted as the ones that die the soonest, are in that group 10 signature. So we walk down the tree this way. Similarly, for every level, we make a decision, and we look at the survivals when we make that decision. So when you look at PI3 kinase group, if you're in that group, you have worse. If you're not in that group, you have a worse prognosis. For heat shock 8, as you walk down the tree, if you're also in that group, you have worse prognosis. And at the very end, if you're in SMAD group 5, you are also having the worst prognosis. So this allows us a quantitative framework for what are protein hallmarks of this particular type of cancer, and a way we can classify patients for targetable therapy. Any questions on this so far? Yeah, okay. So I told you we wanted to go back in the lab.
There seems to be like a group 7 that's in multiple branches.
Group 7, yeah. Yes, here on the. Why are there more choices there?
Yeah.
So because there's still information that can be gleaned from subdividing as you go further. So this one, the only correlative information beyond SMAD5, if you try to add in another group, it's not more informative for classifying the outcome. And in this particular one, as you add differentiation, it is more informative to the others. And this is a paper that is it should be coming out soon. I'll send you the preprint and on the details.
Groups, which exclusive groups?
Right now, so the way the binarization for the patients are going, they are. So if you go back here, it's not fuzzy clustering. It's you're in, yeah. Yes, but there's nothing stopping you from doing that. It's just we chose our first approach would be you're in one of them.
did it by like a random forest kind of classifier? Or?
Yes, a CART analysis, actually.
Yeah.
So.
Quick question on the Cox model.
Yes.
So when you say that the proteins are independent of the cytogenetics, did you run a bivariate Cox analysis to see that each of them contributes statistically?
Yes. So we were asked that from reviewers. Yes. So yeah, no, it's no. Well, no, it's a good question. It's a good question because what we wanted to do is ask the question of, well, right now the state of the art is to classify patients by cytogenetics. Are we adding to that by this information? Okay, so what we also did is we wanted to make this translatable to the clinic, and it's very difficult to work with these rare samples in the lab and do any manipulative experiments. So another thing we did is we ran arrays on hundreds of different cell lines, and we looked at, okay, how can we compare those cell lines to the patient data? And so this is just showing you just principal component analysis or dimensional reduction of all of the 232 proteins that we looked at for the cell lines versus the patient cells. And you can see just looking at it this way, the cell lines don't overlap very much, at all. But looking at it this way doesn't get you the details of what is the same. So we looked at it another way, in that we looked at which are the functional patterns that are recapitulated in the cell lines and across all cell lines. So that allows us to pick out the cell lines which we could actually use to test drugs on certain functional groups and not others. And so that's what we were doing here. So for adhesion and STP, you can see across all of the functional groups, we don't have any similar model system in the cell lines. Alternatively, if you look at some other pathways, like the Wnt signaling pathway and ubiquitin and PKC and FLY1, all of the cell lines can recapitulate those functional groups. So if you want to try a drug that would target those pathways, you would be able to do that for this patient cohort that we're looking at and similar patients. But if you wanted to target adhesion or STP, the cell lines would not be predictive, very likely, of how your drug would respond clinically.
So was this doing like a PLS kind of analysis where you look at the cross-correlate covariance?
What we were looking at is whether, so we did the same type of analysis on the cell lines as we did for the original patient groups, and then we asked what categories do the cell lines fall into.
Oh, okay, but you didn't do like a cross PLS in which having the functional group as the joint covariate.
We're looking at, so as it a classifier for
regression.
We asked whether, so again, what we did was we asked whether across the, no, in answer to you, in answer, but I'll share with the details with you in a second. What we did is we also made the overall pathway analysis available for people to use online. So the whole meta-galaxy approach, from the classification, the cluster optimization, and the network, is available on our Shiny program. So you can drop in data as long as you have some outcomes, whether it's survival or, for you, response of a cell line to a drug. You can import your data and have it be analyzed quickly through this method. We also developed a lookup table or online portal for each of these protein groups to be a resource for the community of people who are interested in leukemia and for people who are interested in the type of protein, or some of the proteins we're looking at in these pathways. So this is also searchable, so you can look at your protein of interest and the patterns of interest in which patients fall into certain patterns this day, and what is the survival for those patients. What we also did with this data is it was independent. We ran a crowdsourced competition a couple of years ago, and my lab worked with our clinical collaborator to make the data set in a more, a less variable package that we put out there for the crowd of people to be able to compete to develop predictive algorithms on. And what they were posed with was saying, given these protein expression patterns and the clinical data, can we predict whether a patient would respond to therapy or not? And so it's a binary question. And there were a whole bunch of models submitted. 31 of them, I have 30 here, 31 of them were rigorously vetted by my lab, and that meant they were performed on a test set of 100 patients, and they were also assured that these models could robustly be run. And when they performed on the test set, what we noticed is interesting in that every model, the average models performed differently than, of course, the top performers and the worst performers. But if you look at them as a whole, they each were informative, because the best performers were on average trying to get good enough. The worst performers were actually, in some cases, overfitting for the outliers or getting the outlier people better than others. So what we thought is, can we take an approach which teachers do, and they use what's called point by serial correlation. Well, some teachers do this when they give a multiple choice exam to people. And you can imagine taking a multiple choice exam, and each question you have a correct answer or not. You can get an A on an exam, and every question you have a probability of getting it right based off of the fact that you got an A overall on the exam. Again, a student who got a D has a probability of getting every question right to a lesser degree. And you can look then across the board of students. Which questions are the students who are overall getting good grades getting right that the students who are not getting an overall good grade getting wrong? And what is the correlation between the grade and that particular question, getting that question right? So we took a similar approach to look at the crowd of models, because we didn't want to fit, we wanted to learn from the whole crowd, and we didn't want to fit for any particular model, because what we realized, it wasn't just the framework of the model, it was the way you handled the data that was informative in part. So what we did is we took the same approach. We looked at the point by serial correlation of every patient in our 100-patient test set, and then we looked at the accuracy of predicting that patient's outcome, and then we looked at the features for all of the patients. And as a reminder, every patient had 40, in this particular challenge, we gave them 40 clinical covariates or correlates, and we gave them 231 protein expression patterns. And so walking through this is just a circuits plot, and the inner part of it, you're seeing the accuracy of the performance for the 100 test set patients around the rim, getting from poor accuracy to the very best as you go clockwise. On the outer rim, the point by serial correlation is shown from negative in brown to green at the very top. And then we looked at the features. So the features in the first group, let me simplify this a little bit. The features in the first group are correlated inversely with accuracy. So if you have that feature, you're more likely to have a poor prediction across all the ensemble models. If you have a prior malignancy, that is one thing that showed up. If a patient had a prior cancer, it's much harder then to predict how they'll respond to therapy. That one might be more obvious for clinicians, but there are a lot of things that were not. Things like the RAS-STAT mutation being positive was actually a positive correlation to a prediction of the outcome. It's in the row 3, or in the third rim here, and that's positively correlated to outcome predictions. So we did this for both the clinical data and we did it for the proteomics data, and it's then giving us features of patients which make their outcomes better or easier to predict, and it's not biasing yourself towards any particular model in the process. So that's the work that I wanted to show you on our goal to be able to quantify their independent features or correlative features of cancer and outcomes to leukemia. Yes?
What was the range? I'm just curious about your different models that were submitted. I mean, you don't want to do this if you have something that's really out of range. Someone who got 100% right.
Yes, no, so good question.
What's the range of the models?
The area of the accuracy curve, so how well overall did it predict patients, was 0.79 was the top model. And the worst model was worse than chance, was chance. But the worst model actually performed well on certain subset of patients. That's what was the impetus for this.
Yeah, but how do you know if it's worse than chance that it didn't perform better on those patients by chance?
No, you're right, because we had them do a scrambled data set as well. And we looked at that. We required them to show us whether they had a feature that they said was predictive or not, and we took out that feature. We did a rigorous testing on whether they were actually using the predictors they said they were using as predictors, and whether it was adding information to their models or not.
If RAS mutation turns out to be a feature of a patient that makes it possible for all models to predict their outcome, does that mean RAS mutation on its own is a predictor, essentially?
So that's what we're trying to tease out right now. Is it predictor alone, or is it a predictor with many other features? And one of the things we're doing is we're asking a head-to-head with clinical predictions. So some of these, what I'm showing you here, are features which the doctor sees anyway and might be able to pick out. And they might be able to say, well, if a certain patient has a RAS-type mutation positive, you would also expect them to have this other feature. So what we started to do is have a clinical survey in which we give clinicians the same data we see here, and we ask them to predict outcomes. And they're not picking up things like, they didn't pick up things like RAS-stat, but they did pick up other features which are correlative or informative on top of what we've picked out. And what we're also going back and is testing this out on a brand new data set of patients to see whether the predictors we thought were predictors here are actually good predictors in the new data set. And in terms of also the question about, well, there might be some models which are so good they're getting some 100% of the patients accurate. That's true for doctors. They actually get, because they're trained to look at cytogenetic categories right away, they group things with patients with T88 transpose transpose 1821 right away off the board, and then they don't classify the other patients as well. So what we've been doing now is using informed models before we go on to the machine learning approaches. So the doctor's rule is guiding certain patients, and then the rest is informed by this sum of the cross-based approach. The baseline models varied. So they had dictionary learning as some of the models. They had regression models. They had, the top performer was using a weighting system of proteins which were inherited more in evolutionary time than others to weight a random forest algorithm. So they are very interesting approaches, but each one contributed in a way which we wanted to look at as a whole. The other part of my work is quite distinct from this in the fact that we're looking at visualizing how cells form tissues, and we're literally watching the cells in these new assays we're doing. And so it's also where the bulk of my new work is going, so I really wanted to focus some time on this work. There's fewer publications because it takes longer to work with these cells, and experiments are time-consuming, but it's one of the most exciting projects we're starting with. And where it came from was actually very personal. My father had a massive ischemic stroke when I was a postdoc studying angiogenesis, or blood vessel growth, at Hopkins. And what was interesting about it from a biological point of view is that my father was very healthy in any way, since the way had no risk factors. It was really unknown. He was exercising and had this massive stroke right after. The day after he had the stroke, the doctors, the neurosurgeons come in and said, you really lost half of your brain, and it's a really bad prognosis. And it did look like that. He wasn't walking, he wasn't moving half of his body. It was really a very severe case of ischemic stroke, a massive stroke. But over the course of a month after rehabilitation, he was able to recover to an extent where he was back at work, he was back exercising, he was playing chess, and by any type of cognitive test, there was no deficit from the point of view of the doctors. They sent him back and said, you don't need any more rehabilitation, which was great. He was still working for a while there, but there were subtle behavioral changes that we saw as a family member, and he also noticed. Things like how often was he on the computer, how often was he talking to people, what was his activity level. These are all things that changed. And unfortunately for my dad, it was a first basically time point of when he started to be into vascular decline or vascular dementia over time. But what I want to tell you about that is that at the very beginning, none of those measurements of his behavior were things that the doctors looked at all or considered. What they also didn't know is anything about what causes a stroke or what is the process of healing after a stroke. And that's what has interested me, and it not is of interest to me, it's interest to many people because of this reason, and that 1 in 5 of us in this room will suffer from some vascular component of detrimental brain health and have something that right now is untreatable, a black box. But behind that black box is this beautiful, elaborate, and very powerful membrane. This is your blood-brain barrier. This is a vascular cast of an animal model of the blood-brain barrier. And every capillary is about the width or less of human hair. And it looks like uncooked ramen noodles, but it's extremely powerful. It delivers 20 to 30% of your oxygen and glucose supply, and it feeds your neurons. So every neuron you can think of abutting or adjacent to one of these blood vessels. And after a stroke, like in my father's case, you see this large infarct or area where you have a lack of tissue, and you have necrosis, you have lack of vessels to deliver oxygen, and you have death of the brain cells in that region. But what I was studying at the time as a postdoc, and I'm still studying, is what the arrows are pointing to, and that's hope, and that is new vessel growth, your ability of your brain to regenerate and repair just like any part of your body. But it just happens to be far less in your brain tissue than other areas. What happens during this process is what's really interesting to me. Can we harness the ability of the brain tissue to self-repair? I don't know if you guys have caught the headline news 4 days ago, I think now, of new work in neurogenesis proposing that perhaps the brain of adults can no longer or cannot have new brain cells present in the hippocampus. That's still a controversial area. It's been so for decades. And the question of whether you can have regenerative properties in the brain matters in the context as well. Are you talking after injury? Are you talking after exercise? Or are you talking after static conditions? And I'm very interested in this process. I'm interested in how we can look at the cells of this process. And the cells, if you zoom into that membrane, that was a vascular cast, so you were only seeing the blood vessels. But the cells are many that make up the blood-brain barrier. They include the capillary cells, which are endothelial cells, which are very, very thin, about 300 nanometers in thickness, that line your blood vessel walls, your microvessel walls. They also include neuroprogenitor cells, cells which can become neurons or can become astrocytes. They include astrocytes, and of course they include neurons. We study all of these cells in the lab. Literally, I wanted to go from our modeling to literally watching what these cells were doing, because then we could test out theories and be able to characterize these cells' properties. So this is a picture of neurons. This is a picture of the endothelial cells we work with. These are the neural stem cells, the progenitor cells, and these are pictures of a growing spheroid of glioma, which are from astrocytes. What we wanted to do is to be able to figure out how these cells move and change and respond through their protein changes in response to regenerative compounds. And for me, it was also a computational problem. We had developed a rule-based model of new vessel growth in the brain. This was basically using game theory to inform a way a cell would respond to other cells in the microenvironment and grow and respond to growth factors. I loved it. It was a toy model which we could use to compare to our in vitro assays in the lab to look at how cells would respond and grow new capillaries in response to growth factors present in the microenvironment. But it was an abstract model or system that characterized the process. It didn't look at the single cells. So we went back in the lab and we looked at the cells that we were studying to look at angiogenesis and asked how did they literally respond to growth factors present in the brain after injury. And we stimulated them with two factors known to be present, brain-derived neurotrophic factor and vascular endothelial growth factor. And then we just watched them. And we fixed and stained the cells and looked at some of the proteins involved in how they would respond, including cytoskeletal proteins, which you're seeing here, which include actin and microtubules and DAPI. But what I thought was going to be an easy computational question was not. We were looking for software that allowed us to tell you that this is one cell and its neighbor is another cell, and they're changing and they're responding in a certain way. I thought we could drop in images, there'd be a way to segment them, and we would be able to get out this beautiful quantitative measure of how the cells were responding to regenerative compounds. Well, there wasn't one, so we ended up building our own, and the outcome of it is being able to classify which are the dominant phenotypes of cells by shape in response to regenerative compounds. And behind this is the tools we developed. So we developed tools, one, to segment the type of cells we were interested in. So this is a modeled and part of adaptive watershed algorithm, where you can look at who are the endothelial cells and their neighbors, and what are they looking like? So these are pictures of our collaborator's cells where he had literally patterned these types of cells. And this is an endothelial cell where you can see the actin fiber alignment, which is color-coded by the angle of orientation of the actin fiber. So that's one metric of many that you can get for these cells. So basically, high content screening for these endothelial cells. But what we noticed is that these cells are not in isolation. They're influenced, of course, by their neighbors. They're changing in shape and structure because of their neighbors. So can we develop a technique where we capture that information and incorporate that as how they're also responding to the drug or compound of interest? So to do that, one of the other impetus or rationale for it is just literally looking at the cells when we were plating them. So you can imagine just adding these cells in, a student comes in and thinks they're plating them at a certain confluency. But when you look at that, say 50% confluency, this example image is a really good indicator of what you might get. You can plate them at the same number of cells, but you're not being able to control how they grow or what they look like at 50% confluency. So that means that a cell that is in the region over here, where it has very little cells around it, is going to respond or be in a different microenvironment than the cell in a very confluent region, local neighborhood. So we needed a method that would allow us to look at the local neighborhood of these cells along with the structure and shape of these cells. So we used a technique that harnessed from graph theory. So this is what we have now online, which is CytoNet. It's a cloud-based program which looks at the spatial analysis of multicellular networks. And we use graphs for multiple reasons. We use graphs in part because it gets a mathematical framework that's linked to the morphology. It allows us to look at things like path length, the number of different connections between cells to get to another cell. You can do it, you can build the graphs multiple ways. We build them right now 2 ways. So after you segment the cells, you can look at the pixel overlap of any 2 cells. You can grow it out by 1, 2, 5, however many pixels you want to, and then draw an edge based off of that threshold you choose. You can also look at just the center of mass and the distance between the center of mass of any 2 cells, and have a cutoff or threshold for the diameter or average diameter between any 2 cells. And then you get a graph out of this, along with the morphological properties. And so for us, that allowed us to look at these endothelial cells in response to different chemical compounds and the regenerative compounds, and have a topological map of how individual cells are responding to the same environment that you put in a single well or a sample. So as an example here, the cells are clustered by their morphological properties into 3 dominant clusters, and by their network properties as well. Each of these cells are representative, or the best average cell visually, of all of the cells in that cluster. So they have, on average, the same length and shape and diameter of that cluster. And what you're noticing is that for us, there's a pattern, of course, with cell size, but that pattern is inversely correlated a little bit to circularity. But what we wanted to get out is these are cells which are subtly changing, and these are cells which are really subtly changing in the brain. Can we tease out a broader mathematical framework to be able to tease out what the network is contributing versus what the drug is contributing to it? So this is what we do through CytoNet. So the paper, which is up in archives right now, will tease out that amount for you. So the network means who are you next to? How many cells are adjacent to you? What do those cells look like? The treatment is what growth factor or compound or chemical you're looking at. And what this is harnessing on is the amount of data in part that you've collected. So you have to be able to have collected enough cells in a certain network property and then treated them across that treatment at the same network property, local network property, to be able to figure out the amount of variance is explained by the network or by the treatment. So for us, an outcome of this is interesting because the mean actin amount, average actin levels in a cell is really informed by the treatment, so the amount of growth factor we give. Whereas for us, the network cells is defining the cell size, the shape of the cell as well. So we would have thought of these as, okay, we give this compound and this is the outcome and the average cell and the structure of the cells, but it's not uniform. And this allows you to look at, across the board, what are the contributions of the network, the local neighborhood versus the treatment. We use graph theory to, yes, go ahead.
How does this change as the cells become more confluent?
So this is on average the contribution from the network and from the treatment. So we look across different types of confluencies. So the network is defined by one of the properties of a network is the number of degrees, so who you're next to. So that's a part of this analysis. So across the board, whether you're next to 2 cells or 5 cells or 6 cells, we're asking, independent of that, is the growth factor having a contribution? Or is the fact that you're next to 2 cells or 5 cells or 6 cells usurping or masking the effect of the regenerative compound that you're adding? So it's a way where right now a lot of this analysis or drug screening is done through high throughput methods where you do high content screening, but you're not looking at the local neighborhood of what's around a cell. And so you have to look at it across the different types of networks, across the confluency, and not just broadly across the Wells confluency, but the local degree of a cell. We use graph theory in a very different way to look at how another cell type in the brain helps in repairing of injury. And here we were interested in a fundamental question of how do nerve cells develop from neural stem cells. And we wanted to approach this by looking at human cells, because human cells in the brain are thought to respond very differently than another organism. Every cell might have a specific type and particular role as we develop. So what we wanted to do is characterize this process in time and quantitatively to come up with a way or a theory of how these cells develop. So broadly, we have a spatial graph, which we're looking at over time as these cells are developing in the lab. And we have a functional graph, which we look at through calcium indicator dye, which gives us a degree or measure of how 2 developing neural stem cells that are developing into neurons are communicating with each other electrically. And then we add in the proteomics, which I showed you in our leukemia application. We're also doing proteomic screening over time for these cells. So what does a video look like? What does an experiment look like? One of the experiments would give us information of a graph, like degree, modularity, meaning are you in a more connected component than the average graph is? And an example that I give broadly is that if you can imagine 2 high schools, and there's cliques in each high school, social cliques in each high school, it's of interest to know those social cliques. Example, social networks look at targeting those cliques through different advertisements. It's equally interesting to look at the people who are most popular in each high school, and then the bridge between it, the person who's communicating between those two popular people at each high school. So these are properties of the graph that we can get also for these cells. We want to know the highest betweenness factor for cells in these developing graphs, because that tells us where's the most information being passed between 2 modules of these developing neurons, as an instance. We also want to know path length. If you've ever played the game of telephone, where somebody is given information on one side of the room and asked to pass it to somebody else on the other side of the room, you know that there can be noise along the way that's added. So the number of people you have to go through, or the number of cells you have to go through here to pass information, matters in terms of the type of information and the speed and the robustness of that information. So graphs allow us to characterize that as well. So this is an example of one of the experiments we do, overlaid with the graph analysis which we do. So these are human neural stem cells, which we plate in a well plate, which is a glass bottom well plate, which is coated with a matrix, which allows us to watch them over days and weeks, and sometimes in cases months. And what you're watching in this particular video over 8 days is that they're developing into immature neurons. Some of the cells are dying, so you can see their nuclei ball up. And in the process, we're looking at edges which are changing and cells which are changing who they're next to. So every frame of this video is processed by the image analysis method that I mentioned. So the soma that are outlined in red are the cells which are alive, which we're using through the graph-based method. And the edges are drawn when cells are within a diameter of each other. We also look at the network through the functional assay, which I mentioned. And here you're seeing the calcium indicator dye lighting up cells which are highly expressing calcium, which is an indicator of their electrical activity. Here, what we do is a different analysis where we're looking at the correlative frequency across images. And we're also looking at which are the cell types that are responsible for that frequency. So here, after we've done a live assay or video, we've fixed and stained the cells for the type of cell they are. So whether they're a neuron that stains for microtubule-associated protein 2, or another marker which stains for immature neurons or stem cells, which is like nestin. Then we do the overlay of the functional image or video with this immunostained image to give you a co-registration of which are the cells that are responsible for which function. From this graph, we can characterize, from these videos, we can characterize the graph, the functional and the spatial graph. So for the functional graph, we just briefly use a method which looks at the identification of the cells and asks the correlation coefficient between any two cells. Is it above expected random or spontaneous correlation between any two cells? There's another step to this in that we also make sure that the cells are spiking at least 2 or at least 3 times during one of these experiments. So as an example, you can see these 3 cells, and by our analysis here, they're correlated above a threshold, so there's an edge of drawn between them. That means their activity is telling us that these cells are communicating with each other. You can see this by their graphs here too, that the spikes in the calcium, or the normalized calcium levels, are also correlated with each other. So then this would be an example of the overlay graph where we have the video overlaid with the immunostain, and then we draw the multiple graph processes here that give us an indicator of what are the cell types responsible for certain features in the graph. So what is an outcome? One interesting outcome that we're noticing is the properties of how these neural cells develop over time. We wanted to be able to come up with a theory, going back to Mendel, it wasn't enough just to collect the data and to observe how these Ps looked. We wanted to understand, can we come up with a seminal property of how these cells are developing? So here I'm showing you some of the first results of this, where you have network efficiency, which is normalized, and it's telling you it's inversely proportional to the number of paths you have to walk between any two cells to pass information in the spatial graph. Over time, as these cells are developing into neurons, we can see that this efficiency peaks at day 6 to 8 in this first cell type we're looking at. And as they're developing into neurons, they're stained for markers of stem cells in the beginning. At the end, they're not staining for stem cell markers, they're staining for a protein associated with immature neurons or microtubule-associated protein 2. That peak also can correspond, or does correspond, with measurements of the electrical activity of these neurons by traditional patch clamp methods. So we were showing that there is a property that's inherent in these cells that is correlated to known functional properties of them becoming neurons. Another thing that's changing over time is the way they're spatially organizing. So in this example, we always normalize for the number of cells. So it's not a property, it's not a characteristic that we include. Every metric is normalized to a random graph of the same number of cells. At the very beginning, you can see they're in these connected components. It's almost like having a community of that's always connected to, all the cells are connected to each other, and they're islands in themselves. By day 6, it's all one connected component. It's like a group of people at a football game, and they're all trying to talk to each other. It's very noisy. But by the end, even in this case without changing the cell number, they're reorganizing into these connected components while they're becoming immature neurons. And as you can see this consistently across the experiments we do, the network efficiency is peaking as they're starting to form this ability to have electrical connections and become immature neurons. And that's inversely correlated to the number of connected components in this spatial analysis. We did this across different cell types, so while the timing changes, the characteristic peak in that spatial network efficiency does not. So when they're becoming immature neurons, you're always seeing this. The number of connected component drops, while the spatial network efficiency peaks. And this also correlates to the active number of cells. So from the calcium analysis, originally our hypothesis was the cells are going to get more and more active as they become more like neurons or more mature neurons. But that's not the case by this particular functional assay. In the midpoint, when they're the most active by the spatial network, when their network efficiency is highest, they also have the highest number of active and correlated cells. That's what we're showing here too. You can see this also by the videos. So at day 1, they're relatively active, but at day 3, they're extremely active in their correlative activity. And by day 5, while they're much more mature neurons, there's very little activity by these assays. Yes?
Did you look maybe to characterize the time scales of the circadian clock? The blood-brain barrier functions differently based on, I guess, time frame in the circadian clock.
Well, so these are human cells, so they're like peas out of the pod. So we don't have measures of the circadian clock, but we do have cell cycle measurements. And so we have transfected the cells for an indicator of cell cycle, and we're asking the exact question that you asked. And I have an example of that coming up really quickly. Let me go through this really quickly. I just wanted to show you what we're doing with this, because where we're going is perhaps just as interesting as the methods that we're developing. And what we're doing is targeting some of the proteins we think are involved in this process and seeing whether it changes the ways these networks form. We're also patterning them. So if our hypothesis is that a certain spatial topography leads to a function, an activity, can we pattern these cells in a way and then ask, can we get out that same function, or do we get out a different function? Do these cells develop networks differently, into neural networks differently? We're also making it patient-specific. There are certain diseases which influence the way neurons develop, and we're focusing on these that are have known genetic components. Why do they develop differently? Are they developing these networks differently from a functional and spatial point of view? What are the processes involved? And finally, linking it to behavior. If we're using this as the peas out of the pod measurement, can we put the peas back in the pod, or at least correlate these peas to the behavior of the organism? So what we're going back and asking is, are these functional assays in the lab correlative to people's, that we're taking the cells from, to their behavior? So for the first, these are the cell cycle indicator assays that we're starting to do. So we transfect the cells with an indicator which tells us which phase of the cell cycle they're in. And these assays, then we also time-lapse assays where we watch the cells over time and we characterize properties of them. One of the first things we were asking is, does it matter how you plate the cells? Because our hypothesis originally was, if they're at 50% confluency but spatially distributed uniformly, they're going to have a different outcome. They're going to develop into neurons differently than if they're localized like they are here. So our first measurements of this are showing that. I'm showing you it where we've looked at medium confluency cells and asked which phase of the cell cycle are they next to, who are their neighbors in this process as the graph is developing. And you can see that the fraction of similar neighbors, they're over time, the cells that are in one phase of the cell cycle here indicated by green, or in another phase of the cell cycle indicated by pink, are tracking in the same way, but then start to become inversely, have an inverse pattern in their relationships over time. But they're all connected to each other. It's not oscillating. Whereas in the low-density cultures, they're oscillating who they're next to, what cell is next to them in the same phase, whether they're next to the same cell, and whether they're next to the cell in the same phase of the cell cycle or not is oscillating at the low-density cultures. So we already know that that matters. Their local neighborhood matters for cell cycle. What we're asking now, does it matter for the function of the cells and how they're developing? It's a really good question to ask. I'm running out of time, so I'm going to go quickly on the other applications. I'm just showing you a little bit of preview of what we're doing. We're patterning the cells in a well where you can keep their environment the same or reproducible over time, and ask, how do these networks form when you do that? Because in the other examples that I was showing you, we would always change the frame of view so we wouldn't over-select a certain region, and they were allowed to move where they wanted to. Here we're restricting them and asking the same questions. Do they develop these networks differently? And we're doing this multiple ways. We're doing it from the neural stem cell stage to the neuron, and also working with a stem cell biologist to go from the original stem cell, human stem cell, to the neural progenitor cell, because the way you would differentiate them through that time point might inform or change the way they act and behave as they develop into neurons. We also are looking at patient-specific applications, so we're working with a collaborator at Sanford Research, Kevin Francis, who published a paper in Nature Medicine showing that children with a disease called Smith-Lemli-Opitz syndrome, which affects the ability of cholesterol to be synthesized, is a disease that also affects the ability of neurons to develop. And in these children, instead of having developed normally as they're in utero, they start to have neural progenitors which seem to develop precociously. or stem cells, which develop precociously into neurons. So our question was, can we go back and watch this process? Can we take cells from these children who have this genetic mutation and culture them and see whether there's differences between them and normal? So that's what we're starting to do, and this is some of the first data that's showing that. So we track over time from 0 to date now, in the 20s, day 26 now, assays we've been doing. And we've also been looking at their healthy controls, which is another stem cell line, which is NL5 here. So the top is the healthy control, and the bottom is the Smith-Lemli-Opitz syndrome cells. And they're stained for markers of immature neurons. At the bottom, it's neurofilament. At the top, it's TUJ. We haven't found necessarily the same trends. This is just preliminary data of precocious development. But what we have found is there's differences, and these differences can be modulated by an inhibitor or agonist of Wnt signaling, which is something that our collaborator had initially hypothesized would be true. What we're trying to do now is characterize these detailed graphs and ask the question of, what is it about the graph in these networks that are changing? But as you can see right here, the segmentation gets harder and harder as you get more and more confluent. So right now, these images are quantitative in a sense only in the early time points, but as they get into the later time points, we're no longer able to track individual cells. And for that, we're working on and are open to suggestions. And these are examples. This is the disease, this is the normal, sorry. This is the disease without any type of therapy, and you can see the cells are different. These are cells that are stained for nestin, which is a marker of neurons, and DAPI, which is a nuclear, and then they're overlaid with the calcium functional graph. There's one cell which seems to be active, and this is over many wells. You can see this example where this one strange large cell that's beating. Whereas if you add in the agonist for the Wnt, you can see that there is a lot more activity. You're rescuing the activity, and the cells are generally more active, and there's more neurofilament. But what's interesting about working with this project with Kevin is that it also brings us to the fact that these are cells from an individual and from patients which are showing behavioral symptoms also. So these children develop, in extreme cases, fatal diseases. So it's fatal very soon after birth or even in utero. Whereas in the less severe cases of the gene, there's just disrupted sleep or repetitive disorders or autism-like spectrum syndromes. So this got us to thinking of, if we want to really extrapolate this work to something at the behavioral level, how do we start doing it? And can we really make this a reproducible assay where we start to collect data from individuals? We take the peas out of the pod and then see how it extrapolates back to the pods or to the people. So that's what we started to do and are starting to do now, where we're getting patients' samples and we're tracking them. So we're tracking them by wearable devices for their sleep patterns over time. So there's a cohort of people who volunteered this last July. We started tracking them now for a couple months or 3 months now. And they're volunteering to also give us cells, which we track in the lab over time as they develop. So are their ability to form these functional networks different? Are they different from individuals? Nobody has actually shown robustly what is the differences across individuals. Is it different if people have different behaviors? This is the same age group of cohort we're starting with. Is it different across ages? These are all things we're asking. And does it correlate at all to behaviors? So one of the first things we're doing is predictions of sleep. So the reason why we're doing sleep predictions is that because one day of lack of sleep, you have changes that are measurable cognitively. And that also includes buildup of A-beta in your brain. This is a particular compound which is thought to be associated with Alzheimer's disease or other neurodegenerative diseases. There's also been proof or shown that your neurons fire a little bit slower if you have lack of sleep. So a few days lack of sleep might be informative for us to think about changes in the brain. So we wanted sleep as a surrogate marker that we could predict over time. And so what we're asking is the question of, can we predict now binary? Is this person going to have a normal sleep pattern or not? And what we're asking people to do is also annotate their data. And they're doing this through an interactive visual system, which we built as part of a startup. And so their data, all their fitness data is downloaded or automatically taken from fitness devices. And then we build computational models of it through machine learning, but we're also asking them to start to annotate the way we're working with their data. And so they can do this by interacting on the screen with their raw data and classifying different data points that are different, like if they were at a conference one day or if they were traveling another day. And why is that informative? It's similarly informative to getting back to what we used for the clinicians. The clinicians pick up features which overall the machine learning approaches might underfit for, that they're not looking for these rules. They're not looking for these outliers. They're trying to get on average as well. But if you have rules embedded, like this person behaves this way when they go off on a conference, those can be used first before you go on to the machine learning approaches. So that's why we're doing it that way. And that's just starting. The other work there's more publications out on it, and we've made a lot of the tools available online. We've tried to make everything cloud-based, because our frustration also in part working with data was a lot of it's not easy to use, a lot of the software that is available, and you guys have already done, you've known that. So hurdles for people using it is part making it user-friendly and not having it user-friendly and not having it available on the cloud. So we've put everything that we can on the cloud. The SetaNet platform is embedded through Amazon, so eventually we're hoping people can store data on Amazon and then just upload it and drop it, or just put SetaNet's folder indicator where they're having their data on Amazon, and it's automatically processed. That gets back to the summary of what we're doing here. Again, it was understanding how cells communicate during processes of growth. And on the first part that I showed you, we were really interested in whether there were these prognostic markers of leukemia. And our work so far has indicated there are these proteomic hallmarks or prognostic markers for acute myeloid leukemia that we can use to classify patients. And what we're right now doing now is high content screening for whether we can show this in cell lines and in patient cells. On the other hand, we've also shown that healthy neurons consistently have a way that they develop in the lab in these assays, that they always have this peak in network efficiency. At the same time, they're having a peak in the fraction of active cells as they start to become immature neurons and electrically active. And so that's something we're seeing as a hallmark of healthy cells, and asking the question of, is it different in people with diseases? And the other thing is, which I probably am preaching to the choir here, but mathematical frameworks underlying the biology and underlying and visualizing the biology can be very informative to understanding biological processes, and for us, cell growth. So getting back to the idea, we're taking the peas out of the pod, our eventual goal is to map what we're doing at the level of single cells in the assay and single cells in the lab in the assays we're doing, back to behavior in the long-term goal. And just to summarize all the people who are involved and contributing, I'd like to thank many of the students and fellows who are working with this. George Britton and Arun Mahadevan have been doing all of the neural and stem cell biology work and experimental work and some of the graph theory. Byron Long's a computer scientist, research scientist, who's been doing the image analysis, along with Wendy Hu, who's been doing a lot of the machine learning algorithms. Cole and Andrew have been working with Arun. Our collaborator, Steve Kornblau, is where we got all of the leukemia data. Kevin Francis has given us the cells from the Smith-Lemli-Opitz syndrome patients, and Aria Warmflesh has been helping us with the patterning. And then NSF has funded this work. So thank you.
Thank you.